On behalf of the Royal Swedish Academy of Sciences, I wish you all welcome to this International Crafoord Symposium in Polyarthritis that is held in honor of the 2013 Crafoord Prize laureates, Professor Peter Gregerson, Lars Kärre, and Robert Winchester. And the motivation for the Crafoord Prize goes for their discoveries concerning the role of different genetic factors and their interactions with environmental factors in the pathogenesis, diagnosis, and clinical management of rheumatoid arthritis. The three Crafoord laureates are a good demonstration that success in science can occur through collaboration rather than competition. Thus, Professor Gregerson has trained with Professor Winchester. They have 13 joint important publications together. And Professor Kärre and Gregerson has collaborated, especially during the last 10 years, and they have almost 30 papers together. So they knew each other when I was phoning them. In the 80s, the Academy received the first donation from Anna-Greta and Holger Crafoord to establish the Anna-Greta and Holger Crafoord Fund that was dedicated to award an international prize annually on a rotating scheme to outstanding scientists in mathematics, in astronomy, in geosciences, and in biosciences. The prize may also be awarded to someone who has made outstanding contributions in polyarthritis, a disease that Holger Crafoord suffered from, especially in his later years. However, a Crafoord Prize in polyarthritis is awarded only after the Academy's class for medical sciences has demonstrated and convinced the Academy that significant scientific progress in the field has occurred. And this took some time, actually. So the first prize in polyarthritis was not awarded until year 2000. And we are proud to have both Sir Ravindra Maini and Sir Mark Feldman here in the audience today. They were the first Crafoord laureates in polyarthritis. Once the decision is taken for a Crafoord Prize, the Academy's Crafoord Committee for Polyarthritis invites scientists from all over the world to nominate candidates. The nominations are reviewed and assessed by the prize committee, and on the basis of the reports from the committee and the medical class, the decision concerning polyarthritis laureates is taken by the Academy in mid-January, the same year the prize is awarded. And then the same evening, I have the great pleasure, it's actually one of the best part of my job, is to reach the laureate and congratulate him. So the Crafoord Prize is truly an international prize. Of the 58 Crafoord laureates over the years, only 2 have come from Nordic countries, and this year, Lars Kärre is the first Swedish Crafoord laureate ever. Keeping with our tradition for the Crafoord Prize, we will not only enjoy the lectures from the Crafoord laureates, but also after lunch, presentations by 4 distinguished scientists, also in the field of rheumatoid arthritis. And we're extremely pleased and honored that you have taken the effort to come here, despite a very short notice, actually, and to share your knowledge with us here at Nobel Forum. So with these introductory words, I hand over the podium to Professor Lars Kärre, who is member of the Crafoord Committee for Polyarthritis. He will share the first Crafoord laureate speeches, and after that, Katarina Svanborg will share Lars Kärre's presentation and talk, and after lunch, Professor Hans Wicksell and Professor Göran Hansson will be the chairman for the symposium in the afternoon. So please, Claes.
So dear Crafoord laureates of the past and of this year, dear colleagues and friends, it's time to open the first session on this Crafoord Lectures and Prize Symposium 2013. And as Staffan Normark mentioned, this first session will be chaired by Katarina Svanborg over there and myself. We will make a brief introduction of each speaker just before his talk. But before that, I have been asked by the Academy to develop a little bit the prize citation. And since we will have separate introductions for each speaker, I will address this task by reflecting a little bit on what is common for all our three laureate speakers that we have today. So we may ask, do these three gentlemen, born in three different decades of the 20th century, share an epitope? Do they have some common features? Apart, of course, from the fact that they are all recipients of this year's Crafoord Award, and also that all three of them actually are trained both as rheumatologists and as basic scientists. Let us begin by looking at the prize citation that was shown by Staffan Normark. Now, a superficial look at the citation may give you the impression that this is handed out as a Lifetime Achievement Award. And indeed, all three laureates do have in common that they would all deserve such a lifetime award, because they have all engaged in long-term, with passion and tenacity, and with the vision to systematically build up data and information resources to link phenotype with genotype, environment, and other factors in polyarthritis disease. But, as the chairman of the Crafoord Prize Committee, Carl-Henrik Heldin, emphasized in his presentation of the prize during the ceremony yesterday, there is much more behind these words in the citation, much more than the daily struggle for a long term with the incremental change and frustrations. Because these words actually do reflect also those magical moments of sudden insight, where confusing and seemingly unconnected observations suddenly make sense and allow you to look at the phenomenon from a completely new angle. And I refer, of course, to that magic moment of discovery that many of you know here. And indeed, the prize awards a distinct discovery where our three laureates share one thing: the credit for bringing together confusing genetics of HLA with an environmental factor, smoking, and bringing them together into a conceptual platform, allowing a mechanistic hypothesis on the pathogenesis of one form of rheumatoid arthritis. And without repeating yesterday's presentation lecture, and not taking too much from the lectures we're looking ahead to, let me just say briefly that what we are talking about, for those of you that were not here yesterday, is of course the discovery that different HLA genes, more specifically for DR beta chains, that predisposes for rheumatoid arthritis, do so because they share a common conformational epitope located in the wall of the antigen binding pocket of the HLA molecule. And furthermore, that this genetic factor, the shared epitope, synergizes with the environmental factor smoking, because smoking leads to modifications of proteins in the lung, citrullination, which leads to triggering of autoimmune reactions, presumably because peptides from such modified proteins bind particularly well to the HLA-DR molecules in question, and possibly also to the relevant T-cell receptors. And we will probably hear more about that in the session, because this is all science still in progress. From the discoveries of the three laureates, a series of pathogenetic events emerge for one distinct subtype of rheumatoid arthritis, which now can be approached in a different way from the point of view of prevention, diagnosis, and clinical management. That is the essence of the prize, shared by all three of you. And finally, I think you share one more factor, one reason for why groundbreaking discovery has emerged from your long-term lifetime struggle. Because the insights and progress that you made could not come only from yourselves and your teams. All good research is in some way based on the collective knowledge produced previously by the scientific community. For example, for the achievement that we are reading about here, we can mention a number of contributions by others. The discovery of the HLA system, the observations of the first HLA disease associations, methodological advances such as serological and monoclonal antibody techniques, cDNA cloning and sequencing, the first HLA crystal structure, the first observations on smoking as a risk factor for RA, and the first observations on antibodies to citrullinated proteins in RA, all of them made by other investigators. So all our three laureates also have in common that they stood on the shoulders of other giants But this is not said at all to diminish your contributions and argue that they depended on pure chance. On the contrary, the hallmark of the outstanding modern scientist is to engage broadly in the scientific literature and community, to harness conceptual and methodological advances made by others and in completely different fields, and apply them to their own field. There is a quotation from one of the world's most famous scientists, Louis Pasteur, that illustrates this. And there are many different translations in English of this, and I chose to present it in Pasteur's original French version. He said, "La chance ne sourit qu'aux esprits bien préparés." In free translation, chance smiles only to well-prepared minds. So by engaging compassionately in your own research, but also by being open to other research areas, our three laureates have in common that they stood extremely well prepared to translate complex, sometimes confusing data and novel techniques rapidly into novel insights and discoveries. Dear laureates, we are extremely happy for that and hope that you will carry on your work in the same spirit so that chance will continue to smile at you for the benefit of science as well as for the patients with polyarthritic and other autoimmune diseases. And we do look forward to hear your lectures. With that having said about the shared epitope, let's now go to the unique epitopes. And it's my pleasure to introduce the first speaker, Robert Winchester. And I think it's fair to say that this is a true New Yorker. He graduated as an MD from Cornell Medical College in 1963. He then went on to do basic research training at the Rockefeller Institute with one of the great icons in immunology, Professor Henry Kunkel, where Robert Winchester developed from fellow to postdoc to associate professor, and where the first serological observations relating to the cross-reactivity between HLA, DR4, and other leads were made. He became a professor and was recruited to the Hospital for Joint Diseases affiliated to New York University in the end of the 70s, where he continued to build his research milieu as a group leader and continued the studies that led to the concept of the shared epitope as a conformational entity in the wall of the MHC class II antigen binding pocket. Since 1991, he's at Columbia University, professor of pathology and medicine at the Division of Rheumatology. He has contributed a long series of discoveries apart from the discovery of the shared epitope in the field of basic immunology, for example, immunofluorescence techniques, basic B and T cell biology, MHC class II molecule expression. He has also made many important contributions in the field of autoimmunity in studies of, for example, autoantibodies, HLA genes, and T cell receptor genes, and in several different diseases, not only rheumatoid arthritis, but also SLE and psoriatic arthritis and celiac disease, to mention a few. He was awarded the Howler Prize of the Arthritis Foundation in 1990, and he has also an extensive record of training fellows and students who later advanced to leadership positions. One example sitting here. And in recognition of this, he has been awarded a prize for excellence in investigative mentoring by the American College of Rheumatology. And the promising title of his talk is Rheumatoid Arthritis and Autoimmunity: Good Genes, Elegant Mechanisms, Bad Results. Please, Robert.
Well, good morning, everyone, and thank you for that very kind and generous introduction. It's an honor to be here, and I certainly offer my profound thanks to the organizers, to the Crafoord Foundation, and to the Royal Academy for making this all possible and selecting us. It's a very humbling experience, and it's, in a respect, a marvelous one, because one looks back into the mists of antiquity in terms of one's life and sees some of these events. My title could very well be the title of many of the talks today, and it's sort of thematic. Good genes, elegant mechanisms, and bad results. But there's a philosophical lesson I think will be increasingly understood as we proceed through the other talks today. I want to introduce to you, and I was at least my assignment was to make the immune system understandable to people who were not immunologists. That might include some rheumatologists. So I had the simple task at first to explain the immune system in a few slides, and those that are experts will have to bear with me on this. And then we'll talk a little bit about the origins of the findings. And I think some of the more current work we'll hear from Professors Gregerson and Klareskog. So let's begin with some definitions of rheumatoid arthritis. First, polyarthritis, as we hear from the Crafoord citation, a disease where there's persistent severe inflammation. It usually involves the small joints of the hands and wrists. But rheumatoid arthritis is a much more specific entity, and we understand it now as a genetically determined and environmentally influenced systemic autoimmune disease. It's initially characterized by the appearance of a strong and broad autoantibody response to citrullinated proteins and peptides called ACPA, anti-citrullinated protein antibodies, and to the constant regions of IgG molecules, rheumatoid factors. And that's the first phase of the disease. Then T cells directed against still unknown peptides play a leading role in initiating the virulent inflammation and destruction of the joint that brings the patient to the attention of the physicians. And we can distinguish phases of the disease. A phase of apparent complete normalcy, where the susceptibility genes activate, are active, but there's no evident disease. A phase when the patient still appears quite healthy, but there's evidence of autoimmunity, the two antibody systems we talked about, but there's certainly no evidence of tissue injury. And finally, and of course there are intermediate stages here, but finally a phase of disease, which is an autoimmune-mediated inflammation that results in the clinical phenotype of joint injury. And of course, there are checkpoints. If we conceive of this as an immune response, the immune response that we're all familiar with, mediated by the adaptive immune system, for example, to a tetanus toxoid immunization, that occurs in a matter of days and is really complete in months, within a month perhaps. But the development of the autoimmune response that we ultimately recognize as rheumatoid arthritis evolves over decades. So there are many checkpoints that are probably inhibiting the development of this horror autotoxicus, if you will. And you can imagine they're parrying and thrusting between different protective mechanisms and different aggressive mechanisms that finally result in a deterioration of self-tolerance into self-recognition and finally autoimmunity. But it's a long-standing process. And then, of course, environmental influences act at multiple sites, and we'll hear much more about that later. So I'd like to begin now in a sense where much of this science began. This is Henry Kunkel, but this is Henry Kunkel on a sabbatical with Professor Arne Tiselius. And really it was this very fruitful sabbatical and its cooperation between these two men that developed the technology of preparative electrophoresis. And Henry Kunkel went on to show that what was then called rheumatoid factor, and in fact anti-nuclear factor, were immunoglobulins. And the paradigm up until that point was that rheumatoid arthritis and lupus were collagen vascular diseases. There was something abnormal in the collagen. But this changed the paradigm from a collagen disease to a disease of autoimmunity with the demonstration of this. So I think it illustrates that the start, the alpha and the omega of much of this began in Sweden. And it's a very fruitful interaction. And of course, I trained with Henry Kunkel, who was a master immunologist. And many of his insights into the serology of immunoglobulins were the intellectual basis of the reactions that we later used to analyze the serology of antibodies against HLA. And of course, Henry described kappa and lambda subtypes, the immunoglobulin subtypes, IgA, and the gene order of all of these. But in doing this, he was a master at serology and recognized that there were shared structures between different immunoglobulins that we call subgroups or kappas and lambdas, and there were individually specific specificities that were later designated as idiotypes. But this concept of serology being individually specific, being group specific, but then having very instructive shared epitopes was engraven in my mind at the level of the immunoglobulin structure, and then we applied that subsequently. So in many respects, this talk really is dedicated to Henry and, of course, his mentor, Theselius. So now some basic immunology. So in the basic immunology, the first thing in orientation to immunology is that the terminology is at best Baroque. So we have this major histocompatibility molecule designated down here in this model from the New England Journal of Medicine Review as a green molecule. And the T cell, the clonotypic T cell receptor that distinguishes each individual type of T cell, recognizes a particular peptide presented in the context of an MHC molecule. And each T cell responds by clonal expansion and differentiation to a memory and effector stage. But the critical recognition is the T cell receptor, the clonotypic receptor, recognizing peptide in the context of MHC. So it's a trimolecular interaction. And we can see it here designated in this pi-mole reconstruction from coordinates in the data bank. Here's a T cell, alpha-beta T cell receptor. There's a bit of the peptide that we can see, but otherwise it's sort of embraced by the T cell receptor. And here's the MHC class 1 molecule in this case. And we see its structure right away. The thing that is striking are these two alpha helical margins of the binding groove and a beta pleated sheet structure that forms the base. It's sort of somewhat biconcave saddle-shaped structure. And so the peptide sits in this groove, embraced really by these, or insinuating itself under the alpha helices and above the beta pleated sheet. And then there's a sort of stalk, another very typical immunoglobulin-like domain that goes down to the surface of the antigen-presenting cell. And this would be the T cell receptor's view of this structure. And the peptide lies in this groove, illustrated here, again with side arms that bind. But in a space-filling molecule model like this, we see that the peptide molecule interacts in multiple ways. And of course, some of the hydrogen bonding shown here in yellow dots points up towards the T cell receptor, but many parts of the molecule are bound into the MHC matrix. It's a very tight and close unit. And then the T cell receptor, as one could have imagined from the Zinker-Nagel-Doherty experiment, interacts with a much larger footprint of the MHC molecule and covers nearly all of the peptide. So again, now we see it with the loops of the T cell receptor interacting with both peptide and with MHC. That's really the central evolutionary event. This process evolved some 400 to 450 million years ago, and both dinosaurs, human beings, mice, chickens, used the same fundamental recognition structure. So let's see some of the challenges, again, to review a bit of immunology and why the MHC is so critical, and then ultimately to lead us to the disease interests that we have. So there are some 10 to the 13 different peptides, and correspondingly you need something on the order of 10 to the 13 different T cells with structurally different T cell clonotypic receptors so that each progenitor can recognize a different peptide. And in order to do this, since there is not enough room in the nucleus to encode enough DNA for this many genes, these genes are generated randomly by somatic recombination. And there's a selection event within the thymus. So the clone bearing a T cell receptor appropriate for the individual in terms of their MHC and peptide structure is included. So the entire system uses self-peptides bound to self-MHC as a surrogate for the pathogenic peptide. So the entire immune system then is built on a recognition of self. And we use self-peptides as a mechanism of recognizing all subsequent pathogens. But if the entire adaptive immune system is built on self, you have a very ready explanation, at least in terms of the theory of allograft rejection, in which the non-self graft looks like a pathogen, and autoimmunity, in which the self looks like a pathogen to the immune system. So that's our sort of philosophical framework. But now the MHC comes back, or the HLA molecules, to anchor each of the 10 to the 13 or so different peptides with high affinity. It's not molecularly possible to do that in a specific way. And it's absolutely essential, because if the peptide elutes from one MHC and binds secondarily to another, that would signify that cell as a target for destruction, which of course would be inappropriate. So there must be a very intimate and irreversible binding of the peptide to the MHC, such as if the peptide leaves, the MHC molecule structurally collapses and becomes very ineffective in terms of its ability to secondarily bind another one. So there must be, almost by definition, different kinds of MHC molecules that are going to bind particular subsets of peptides. And indeed, that's exactly the case. And there are two general mechanisms to enable the wealth of all of these different peptides to be presented. And for the individuals, one duplicates the genes of the MHC. So one has HLA-A, HLA-B, HLA-C loci, and the same with HLA-DR. For the species, one develops alternative gene forms, or alleles, that encode MHC molecules with different peptide binding properties. And this is a system, as perhaps the numbers here reflect maybe about a 6 months ago level. But there's no region in the human body that is obsessively endowed with the number of alleles that we see within the MHC. So at the HLA-B locus, 2,862 alleles. At HLA-A, 2188. Not all of these bind different peptides, but the vast majority of them are distinguished by differences in peptide binding. And much the same with the three HLA class II loci, DR, DQ, and DP. And if, and it is not the case, that these were all independent, one ends up with a multiplication of these three numbers into a salary that, well, is more than that of any conventional immunologist. And the same with HLA-DR. And then if you multiply these two numbers together, and I was just unwilling to do that, but you can see the number in principle, the immense diversity. So this equips the species with a large number of alternative polymorphic MHC molecules. And what one has here is frequency-dependent selection and heterozygote advantage. So basically, the individual with the least common MHC structures is the person at the greatest advantage, let's imagine, in the case of an epidemic that has somewhat selected for a predominant type of MHC molecule. And heterozygous advantage is the person with more different structures for binding different kinds of peptides has an advantage over a homozygote. One can also imagine that there's a social context and that the pluralism of different people, so heterozygosity and cherishing the exception, the unusual person, is very much the same, something we do as a human race, or I hope we do as a human race. And here it's done again by biology in terms of the HLA or MHC system. So let's talk just a little bit more, and then we'll get on to the science. So again, a very nice article in New England Journal. These are the different peptides, and I've just selected here for its simplicity HLA-B7 and HLA-B27. And the rheumatologists in the group know that HLA-B27 is associated with a particular autoimmune disease. But this is the peptide that has the proclivity to bind to the HLA-B27 molecule. It's got an arginine at position 2 and a lysine at position 9. HLA-B7 molecules bind a different set of peptides with a proline at position 2 and a leucine at position 9. So the repertoire of peptides are selected by these two molecules. Well, what does this mean? So this is a slide I prepared for the medical students, but it illustrates the point well here. So if we look at HIV envelope proteins, and we look at the number of peptides in that envelope that can bind to an HLA-B27 molecule, and here in a little more detail is the total binding motif, arginine, that can be lysine, arginine, tyrosine, or leucine. There are some 15 different peptides in HIV envelope that will bind to B27. HLA-B7, relatively fewer, 5, have the proline leucine motif. And in the case of HLA-B35, proline, tyrosine, there are no peptides. Well, does this correlate with anything? So long-term non-progressors, people that in the days before there were highly effective antiretroviral therapy for HIV, if you're HLA-B27, your mean survival to the development of AIDS was 14 years. If you were HLA-B35, your mean survival to the development of AIDS was less than 2 years. So clearly, the way, the number of peptides that your MHC binds in the case of a pathogen importantly influences your host. There are other factors, it's far more complex than this, but this illustrates in a simple way the difference that people have in terms of recognizing the number of peptides that are present in various pathogens. So perhaps that's also an explanation for why B27 is associated with an autoimmune process, simply because of that. There are other explanations too that may apply, and we don't know the answer. So let's then proceed more quickly. So during development of the adaptive immune system, MHC molecules bind different self-peptides. They select T cell clones with clonotypic receptors that recognize only the individual self-peptides that are bound to their own molecules. And some of these self-peptides selected by certain molecules may become the target of subsequent autoimmune responses. And this accounts for the fundamental association of certain HLA diseases with autoimmunity. The search for these particular molecules is still an active and ongoing process. But because of the allelic diversity of the MHC, each individual is nearly unique in terms of their peptide binding properties. So we have these two elements, a related sharedness that groups of people have because of the property of their alleles, and the ultimate uniqueness of each person. So now let's go back in the midst of history and go into the 1970s, when the concept of the HLA region, the MHC, consisted of 3 loci, HLA-A, B, and predictably in immunology, C is located in between A and B. Now, immunology itself, the immune response, let's say compared to cardiology where the heart beats, or pulmonary science where the lung moves, not much happens in the immune system. So some of us were fascinated with the mixed lymphocyte culture because one could take lymphocytes from one person and mix them with lymphocytes for another and watch under a microscope after a few hours, and these cells begin to recognize one another. So it's a very dynamic and fascinating process, and made all the more interesting to those of us in autoimmune disease by a finding back in 1968 by Ralph Williams in Astorga, showed that rheumatoid arthritis patients were mutually unreactive in an MLC. So there's something about MLC responsiveness and rheumatoid arthritis. They did not interpret this as genetic. They thought it might be a disease manifestation. We were also very interested at that time, the lupus patients also have major defects in their immune responsiveness, particularly in the recognition of the mixed lymphocyte culture reaction. And lupus patients' sera inhibited this. So we wanted to look at the question of what was going on in these two autoimmune diseases with the MLC. But we also included a control, and that is certain pregnancy sera that inhibited the mixed lymphocyte culture reaction, but lacked conventional cytotoxicity for these known antigens encoded by these genes. So what would that look like? And the answer was this, that it was preferential staining of B cells, here a B cell line, here a T cell enriched preparation, but there's a B cell, and here a B cell enriched preparation. And the thing that made this reaction possible, or recognition, almost at the time or a little bit before this time, it was felt that B cells had surface IgG as well as IgM because they were all positive. But using fluorescent reagents that were Fab prime 2 fragments, one could avoid any of the binding of that. And so a B cell would appear in indirect fluorescence, the nature of what this reaction was, as entirely negative. So then one could show with great degree of confidence and a high degree of specificity that pregnancy sera, indeed, certain pregnancy sera reacted with B cells. And this is a system that we now call the HLA-DR system. And then this was applied. And the concept then, driven largely by considerations in the field of HLA typing, was to find typing sera and to type autoimmune diseases. DR4 was significantly increased in rheumatoid arthritis, as we and Peter Stastny showed, but of course not in SLE. And here's SLE samples. And notice the number 24 patients or 44 patients. Yeah, life was very simple in those days, and with just a few dozen patients, and in your own clinic, one could show the difference, the significant difference between rheumatoid arthritis. But there were certain sera that were not at all good typing sera, but that had a preferential reaction. Here's a particular serum called 1283 that detected 80% of patients with rheumatoid arthritis and only 12% of patients with lupus. So these sera were not of much interest to the HLA typers because they didn't give specific defined results. But for those of us of a rheumatology persuasion, we were fascinated with what this serum identified that was common among different patients with rheumatoid arthritis. So there seemed to be, as I said, the DR4 association strong and reproduced in New York and London, in California, a strong association. And DR1 was weakly associated. But if one went to Jerusalem, essentially if you cross the Bosporus, the association changes. And so in the Eastern Mediterranean and on through that portion of the world, the association with DR4 is far weaker and its association with HLA-DR1. So it really suggested the possibility, well, two possibilities. One, that the HLA system bore no rational relationship to disease and it was just some happenstance of different ethnic groups. Or the position that we took, that some variety of HLA genes were associated with rheumatoid arthritis while other varieties were not. One didn't know because one at this point had only serology that in fact these were different genes, different alleles. And that certain other HLA-DR genes such as DR1 were associated with rheumatoid arthritis. So the concept then was that one could put this story together. Now, using human pregnancy sera, it was a difficult and complicated and less than persuasive. So a very important strengthening concept was the identification of a monoclonal antibody that had a very similar set of reactions. I won't get into the subtle details between the epitope recognized by this particular monoclonal antibody and the concept of the shared epitope, but it's in those publications if anyone wants to follow. So the hypothesis then was that there was a structural feature shared between some HLA-DR1 and DR4 alleles or molecules encoded by those alleles, that certain pregnancy sera recognized call a shared epitope, and that this structure was absent from DR4 molecules in populations where DR4 was not associated with rheumatoid arthritis. So what was the molecular basis of the shared epitope structure recognized by these selected sera? So enter Peter Gregerson at this time. And here we were, aspirin pushers, as Peter calls us, trying to deal with this puzzle of could there be a molecular basis to this phenomenon? A molecular basis that did not really fit into the paradigms of HLA typing, which were much more driven by allelic specificity. But we had sat together and plotted just how one could do this. Well, at that time, of course, nowadays, any one of you could go into the laboratory and kind of reproduce all this with PCR reactions, and you'd call up your distributor and you'd get kits to make cDNA and reverse transcribe, and you'd have a computer that had a nice screen and you could align sequences and you'd have an automated sequencer. At that time, none of this existed. And the concept of even what cDNA is was a bit foreign to we rheumatologists. But we knew that we really had to sequence the genes. And in order to do that, we invited Jack Silver, who is a very good molecular biologist, to participate. And this is basically what, and I should say it took Peter about a year, if that's fair to say, to sequence this one. Well, now we can do this in an afternoon or in just minutes on a new generation sequence, but it took him fully a year to sequence this. And the result of the first two patients, one selected or representative cell lines, in this case, one from a rheumatoid arthritis prototypic allele, another from a DR4 not associated with rheumatoid arthritis, and in DR1. So DQ beta identical, DQ alpha identical, DR beta nearly identical in the case of these two fours, but with one little difference here. Now, at this point, there was no sequence alignment program that one could look at and put these sequences. So the sequences are written out in pencil on long sheets of paper. And one kind of was like a biblical scholar with these scrolls, and they'd go on for many feet. And this is how you did your alignment. And then there was no computer that you could get the translation. You had to write it all down step by step. So to our very great amazement, the concept was, well, this had a structure here, a sequence, a nucleotides and an amino acid sequence. It was absent here, but present here. And so after a little bit of thinking about all this, Peter wrote this paper, The Shared Epitope Hypothesis, that tried to explain what was going on. And basically, what was evident is that there is this patchwork structure. Here's the motif in DR4, LQKA for 0401. So lysine at this position, 0404, a different DR4 allele, but also associated with rheumatoid arthritis, arginine, where the 0402 not associated had two negatively charged amino acids, aspartic acid and glutamic acid at this point. And indeed, the same LQKA or LQRA motif was found in DR1. So that this we defined as sort of the shared epitope region. We still didn't have a very clear notion of what this epitope meant, and we tried to construct some molecular diagrams and got it, of course, all entirely wrong where this might be. But as we'll see subsequently in X-ray crystallography studies, it turned out to be a binding pocket. But before that, when asked, well, the shared epitope hypothesis was developed to explain a pattern of association between DRB1, and it did that, but did it have predictive value? So there are two very interesting confirmations of this as a hypothesis. And one is Yakama Indians, who are the Paleo-Asians that came across from northern Asia across the Bering Straits about 16 to 14,000 years ago. And then these constitute the Native American Indian populations. And they're very interesting in terms of their subgroup and their proclivity of some of these nations to have very high frequency of rheumatoid arthritis. So the Yakama Indians have about a 5 to 7% frequency, population frequency of rheumatoid arthritis. And they're characterized by a particular HLA allele of DR6. You see all of the terminology, which we won't go through, but it's DRB1 1402. And this is, in fact, contains the shared epitope motif. 83% of the Yakama Indians with rheumatoid arthritis, but in 60% of the controls. So a genetic bottleneck in this group selected a very unusual HLA allele, which happened to have the shared epitope, work of Wilkins and his associates. And then exploring the MHC type of RA cases positive for the serologic shared epitope resulted in the identification of HLA DRB110, which was at that time a new allele that did in fact have the susceptibility, had the shared epitope sequence. And Takami Matsuyama demonstrated that by immunoprecipitation studies and sequencing. So it seemed to be predictive. So were the alleles then identical to wild-type alleles? And at this point, sequencing became a bit easier, and Parvin Merriman and Peter together sequenced the alleles in an RA patient and showed that, in fact, these were the wild-type alleles, and that was the first time that actually sequences from a patient was done. The other were prototypic cell lines. So these are good genes. And the concept of horror orthotoxicus, the seemingly abnormality of rheumatoid arthritis or autoimmunity in general, really needs to be evaluated now, because the susceptibility genes were no longer genes that encoded some pathologic protein that seemed to be different, but this was the normal composition. So it's something far more subtle and far more physiologic that is going on. So we mentioned the concept and the importance of the crystallographic details, because now it's evident that the structural region along this alpha helical margin of the MHC class 2 HLA-DR molecule at position 70 here, 71 the position of arginine lysine, 74 alanine. But the remaining amino acids of this region really defined a very critical binding pocket that bound shown here in the diagram, the central pocket that binds the so-called P4 side chain of the peptide in the MHC molecule. And the electrostatic bonds between this positively charged arginine or lysine, in this case, are illustrated with this representative peptide in here, showing the effectiveness and the importance of the hydrogen bonding between this element of the shared epitope motif and a negatively charged side chain of this peptide. And then in the case of where at position 70 could be in some alleles an aspartic acid, and position 71 a glutamic acid, as is in the case of DRB10402, that hydrogen bonding is no longer possible. So that these shared epitope negative molecules really had a fundamentally different type of peptide that they would bind. So one of the concepts, and this will be dealt with at much greater, but this is the work of Bell and Hill back in 2003, showed that peptides modified by citrullination, and you'll hear much more about that, can bind to HLA-DR molecules. And this was the first demonstration of that. Now, whether this is relevant, we're going to leave to the next two speakers. But here's an example of their work, that shared epitope alleles did not bind the wild type or arginine containing vimentin peptide. But when that arginine was modified to citrullinine by the loss of that amino group, then this uncharged molecule at this point bound effectively to these three shared epitope molecules encoded by these alleles, and did not preferentially bind to other non-shared epitope containing molecules. So it's a very important clue that may point a direction. But the risk for rheumatoid arthritis is really defined by the individual's genotype, so it's much more than their particular haplotype. And it's the number and type of HLA-DR molecules. And a very good example of this is in a recent publication from Rodier, who's summarized, I think, in a very elegant way. So here are a group of different alleles, the same group shown here, shared epitope alleles. Here, these are O4 shared epitope. In their terminology is O404, O405, the form of DR4 that's common in Asians, and O408. And there's O401, there's DR10, there's DR1. And here are the same alleles here. So the relative risks of developing rheumatoid arthritis, the genotypic risk, in the case of a DR10, O401 compound heterozygote person is 28.2. And the risks vary. So it's a function not only of the generic shared epitope, but the particular kind of allele has an influence. And then if you have down in this group where there's a single shared epitope, varying lower degrees of association, and over here where shared epitope negative and shared epitope negative have negative relative risks. There's no absolute protection. There's just a variety from risks of 0.2 as low as 0.2 up to 28.2 across this. So a vast difference in genotypic risk, but nonetheless the shared epitope structures seem to, when present in a double dose, either in a true homozygote or in a compound heterozygote, confer a much greater risk. And again, we'll hear a lot more about that. So I think that's about where I want to conclude. The risk for the development of rheumatoid arthritis is defined by the number and type of HLA-DR alleles that together encode the shared epitope binding pockets versus other types of binding pockets. And this suggests the possibility that during establishment of the repertoire of T cells, the binding of particular self-peptides by DR molecules that contain pockets defined by the shared epitope selects a T cell clonotype critical to the development of rheumatoid arthritis. And this establishes the basis for an adaptive immune response directed towards one or perhaps a few still unknown peptides in a target tissue that ultimately results in a disease. And the concept that this is a Cinderella's slipper, so we're in the place of Prince Charming, knowing the structure and having this as a probe to use for the peptide that might ultimately bind. And we can just conclude by seeing within the area of genetic susceptibility. We're going to learn far more about this from the next two speakers. Interdiction of this process we'll hear later in terms of mechanisms that we can thwart the process of joint injury, and we'll hear about environmental interaction. So within this diagram, many of the later speakers are going to fill in the gaps. Thank you very much for your attention.